Good day to all. Now we are going to see about scientists and their discoveries. 12th standard biozoology book, important 1 mark. In this, all the scientists and year what they have discovered is explained. The parthenogenesis was first discovered by Charles Bonnet in 1745. Sex hormones were discovered by Adolf Puttenant. Carl Landsteiner discovered two kinds of antigens called antigen A and antigen B on the surface of red blood cells of human blood. The fourth and the rarest blood group, AB, universal recipient, was discovered in 1902 by two of Landsteiner students, Van de Hazel and Stirling. Bernstein in 1925 discovered that the inheritance of different blood groups in human beings is determined by a number of multiple allelic series. The Rh factor or Rh antigen is found on the surface of erythrocytes. It was discovered in 1940 by Carl Landsteiner and Alexander Wiener in the blood of rhesus monkey. Mary Lyons suggested that bare bodies represented an inactive chromosome which in females becomes tightly coiled into a heterochromatin, a condensed and visible form of chromatin, Lyons hypothesis. Tiggio and Levin 1960 described a simple method of culturing lymphocytes from the human blood. The concept of the gene was first explained by Gregor Mendel in 1860s. In 1909, the Danish biologist Wilhelm Johansen coined the term gene that was referred to as discrete determiners of inherited characteristics. According to the classical concept of gene introduced by Sutton in 1902, one gene, one enzyme hypothesis, the experiment job George Biddle and Edward Tatum in the early 1940s, and Neurospora crassa, the red blood mold, led them to propose one gene, one enzyme hypothesis, which states that one gene controls the production of one enzyme. As early as 1848, Wilhelm Hofmeister, a German botanist, had observed that cell nuclei organize themselves into small rod-like bodies during mitosis called chromosomes. In 1869, Friedrich Misser, a Swiss physician, isolated a substance from the cell nuclei and called it as nuclein. The nuclein was renamed as nucleic acid by Altman 1889 and is now known as DNA. Oswald Avery, Colin MacLeod and Maclean McCarthy in 1944 repeated Griffith's experiments in an in vitro system in order to identify the nature of the transforming substance responsible for converting a non-virulent strain into virulent strain. In 1952, however, the results of the Hershey-Chase experiment finally provided convincing evidence that DNA is the genetic material. Alfred Kersey and Martha Chase 1952 conducted experiments on bacteriophages that infect bacteria. Based on the X-ray diffraction analysis of Maurice Wilkins and Rosalind Franklin, the double helix model for DNA was proposed by James Watson and Francis Crick in 1953. Frankel contract and Singer 1957 first demonstrated that RNA is the genetic material in RNA containing viruses like TMV, tobacco mosaic virus, and they separated RNA from the protein of TMV viruses. Dufra 1965 proposed a single standard model UNINEEM as a long coiled molecule which is associated with histone proteins in eukaryotes. Semi-conservative replication was proposed by Watson and Crick in 1953. Marcel Nirenberg Swaro-Ohava enzyme polynucleotide phosphorylase called Ohava's enzyme. Hargobind Khorana, Francis Crick, and many others have contributed significantly to decipher the genetic code. Hubble hypothesis. It is a hypothesis proposed by Crick 1966. Jacob and Monod proposed the classical model of lac operon to explain gene expression and regulation in E. coli. In lac operon, a polycistronic structural gene is regulated by a common promoter and regulatory gene. The DNA fingerprinting technique was first developed by Alec Jeffreys in 1985, recipient of the Royal Society's Copley Medal in 2014. Oparin in 1924 suggested that the organic compounds could have undergone a series of reactions leading to more complex molecules. Haldane 1929 proposed that primordial sea served as a vast chemical laboratory powered by solar energy. Haldane coined the term prebiotic soup, and this became the powerful symbol of the Oparin-Haldane view on the origin of life. 1924 to 1929. Urey and Miller 1953 paved way for understanding the possible synthesis of organic compounds that led to the appearance of living organisms. Jean-Baptiste de Lamarck was the first to postulate the theory of evolution in his famous book Philosophie Zoologique in the year 1809. Lamarck's theory of acquired characters was disproved by Auguste Weismann, who conducted experiments on mice for 20 generations for cutting their tails and breeding them. The followers of Lamarck, neo-Lamarckists like Hope, Osborn, Packard, and Spencer tried to explain Lamarck's theory on a more scientific basis. Charles Darwin explained the theory of evolution in his book The Origin of Species by Natural Selection. New facts and discoveries about evolution have led to modifications of Darwinism and is supported by Wallace, Heinrich, Haeckel, Weismann, and Mendel. Hugo de Vries put forth the mutation theory. De Vries carried out his experiments in the evening primrose plant, Aenothera lamarckiana, and observed variations in them due to mutation. According to De Vries, sudden and large variations were responsible for the origin of new species, whereas Lamarck and Darwin believed in gradual accumulation of all variations as the causative factors in the origin of new species. Swalwright, Visser, Mayer, Huxley, Dawsonski, Simpson, and Haeckel explained natural selection in the light of post-Darwinian discoveries. In the 1950s, experiments by Porter and Edelman revealed the basic structure of the immunoglobulin. Dr. Edward Jenner prepared first vaccine for smallpox in 1796. Polio vaccine was developed by Dr. Jonas Salk. Vaccine consists of inactivated microorganism. Dr. Albert Sabin live attenuated oral polio vaccine. Louis Feist 1885 discovered vaccine against rabies, anthrax and cholera. PCG vaccine was developed by Calmet and Guerin against tuberculosis in France in the year 1908. Selman-Waxman discovered streptomycin and was the first used the term antibiotic in 1943. The first antibiotic discovered by Alexander Fleming in 1926-49. Penicillin is also referred as the queen of drugs, and its full potential as an effective antibiotic was established much later by Ernest Chain and Howard Florey when they treated the wounded soldiers in World War II with penicillin. Fleming, Chain, Florey were awarded the Nobel Prize in 1945 for the discovery of penicillin. Best and Banting in 1921 isolated insulin from the pancreatic islets of a dog and demonstrated its effectiveness against diabetes. Interferons were discovered by Alec Isaacs and Jean Lindemann in 1957. ELISA is a biochemical procedure discovered by Eva Engvall and Peter Perlman in 1971 to detect the presence of specific antibodies or antigens in a sample of serum, urine, etc. 54. The polymerase chain reaction (PCR) is an in vitro amplification technique used for synthesizing multiple identical copies, billions of DNA of interest. The technique was developed by Carey Mullis, Nobel laureate 1993 in the year 1983. Ian Wilmot and Campbell removed 277 cells from the udder of an adult sheep and fused those cells with 277 and fertilized egg cells from which the nuclear material was removed. Charles Elton 1927 was the first to use the term niche as the functional status of an organism in its community. Want half proposed that with the increase of every 10 degrees Celsius, the rate of metabolic activity doubles or the reaction rate is halved with the decrease of 10 degrees Celsius. This rule is referred as the one-halves rule. The term biodiversity was introduced by Walter Rosen 1986. German naturalist and geographer Alexander von Humboldt explored the wilderness of South American jungles and found that within a region the species richness increased with increasing area but up to a certain limit. G. Namalwar was a supporter and expert of organic farming. Dr. Sultan Ahmad Ismail is an Indian soil biologist and ecologist from Tamil Nadu. Thank you, student.